Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For the section to discuss NCERT Class 12, the chapter Biotechnology and its applications. Part 3, Biotechnological applications used in medicine. The recombinant DNA technological process have been that made immune impact the era of healthcare, enabling massive production of safe and more effective therapeutic drugs. So in this biotechnology applications, we are using as an medicinally more and more when your production of as an the number of medicines and effective therapeutic drugs we are obtaining from only for this biotechnology with help. Next, further recombinant therapeutics do not induce unwanted immunological response. In common case, the similar products isolated from as a non-human source. So some unwanted things also we are completely to get removing with help of in this biotechnology. Next, at present about 30 recombinant therapeutics have been approved human using world over. So mostly 30 very important using 30 recombinant therapeutics we are approved for as an entire over a world war. In India using as in 12 these presently you beginning in market. In India 12. In India 12 the presently beginning in that market. Okay. Next, first one we discuss genetically engineered insulin. Genetically engineered insulin. First, management of adult one set of diabetes possibilities takes taking insulin at regular time of intervals. So any one person to get causes as any that diabetes. What happen? It's every time in particular interval time period insulin must and should is taken. Now, what would a diabetic patient do not enough the human insulin was not available? If in case human insulin is not available, what happen? Automatically the death is takes place simply. Okay. If you disease, you can would be the soon realized that one would be have the isolated from insulin from as an animals. Once you think that same kind of insulin to get obtained from animals, would the insulin isolated from other animals just the effective that secreted by as in a human body itself would be there not their immune response to the human body. So sometimes we are uptake that is good, but every time we do like what happened it is sometimes they are get as a result, sometimes they get give as any some bad things. Okay, now imagine the bacterium where they are available that is could make human insulin. Suddenly their whole process become there simply. You can easily to get grow large quantity of bacteria is make much insulin unit. So let's once imagine if in case the bacteria is produces a number of insulins, that is to get how much quantity of insulin we require easily to get uptake with the help of the bacteria. That is the better I thought. Next, think about where the insulin can be there orally administered to as in a diabetic people or not. Why? So mostly already that uh, we are insulin we are uptake from that animals also in slaughtered pigs and cattle. Am I right? Let's pancreas mostly. Yeah. Insulin used for as a diabetic patient was earlier extracted from pancreas of selected cattle and pigs. So when they kill to as in that pigs and cattle, we extract from as any what insulin the presence of in the pancreas contain as an insulin. Am I right? Yeah. Insulin from animals source through as an cause there completely caused source of patient develop as allergies and other types of reactions and for going the protein. So mostly sometimes we are directly inject to as in this insulin to as in a human. What happened to get causes as a number of diseases. Insulin consists of two types of short polypeptide chains, that is chain A and chain B. So insulin mostly they having as in A, there are two polypeptide chains. One is A and B. It is linked together in disulfide bridges. Here they are complete to get bind with as in a disulfide bridges. Disulfide bridges. See here insulin this is in A, is a A, it is in a B. So between these to get making as what here disulfide bridges will be a coming. Disulfide bridges. In mammal, including as in humans, insulin they synthesized a pro-hormone like proenzyme, like as in a pro-hormone. It is also need as in that produced before becomes full matured functional hormone. So in this once a and B chains after to get mature, it is to get make as in a completely full mature. Then what happen? Which contain extra shade is called as in a C peptide. So finally, after get making as a disulfide, it is make as what in a C peptide will be to get formed. See, this C peptide is not present in the mature insulin and removed during the maturation into as insulin. After get matured, then what happened? It is to get 30 in these different regions. It is make as in a complete mature insulin. Next, the main challenge for production of insulin using as in a recombinant DNA technologies was getting insulin assembled into as in a mature form. So when this total process we are obtaining from is an a recombinant DNA techniques. We are getting easily the DNA we are uptaking. See, mostly in that in 1938, Eli Lilly American company. Eli Lilly American company. It is prepared two DNA sequence corresponding to A and B. So in this 1983, the American company, even Lilly, it is to develop as an A and B two corresponding DNA chain. The human insulin was introduced into that in a plasmid E. coli. So it is to get directed to introduce into as well in a Escherichia coli. Now it is produced insulin chains. So the insulin, the mostly the E. coli to get produced as what? Number of insulin chains. Let us chain A and B where they produced separately. A and B, A and B to get developed as in a separately. Extract is a combined with as in creating their their completely disulfide bonds with a form as in a human insulin. So in this A and B stands to get developed as in a new additional DNA also to get developed at C type with the help of in a disulfide bonds. Okay, now this is in completely in a human insulin. Gene therapy. Next we discuss gene therapy. A person born with as an heritability disease can correct therapy for taken such as in a disease. Any person to get as any hereditary disease, how to get control. See, gene therapy is attempt to do this. Gene therapy to attempt to do this. The gene therapy is in a collection of methods that allow in collection of a gene defect as any has been the diagnosed child or embryo condition. So in this disease, we can easily early to diagnose as any in child or embryo condition. Here the genes are inserted into as in a person's cell and tissue to the complete the treat a disease. So any genes or any cells to get introduced into as an a the human cell body and to get completely to get cure this disease. Next, corruption of the genetic defect involves the delivery of normal gene into the individual or embryo. It's take over the functional and their state for as a non-functional gene. So here which genes that harmful, that genes to get they completely to get modified and further to get complete to control to as in a this disease. The first clinical gene therapy was given in 1990 in 4 years old girl with a adenosine diaminase ADA deficiency. So in this 1990, in 4 years old baby with as in a cause of the disease ADA disease, that is adenosine diaminase. This enzyme is crucial for the immune system to function. This is very important for as an immune system function. This disorders caused due to the deletion of the genes, that is adenosine deaminase. If in case why this disease they comes, because diaminase adenosine diaminase this is their absence. The some children's ADA deficiency can be there cured with bone marrow transplantation. Other can treat the enzyme is enzyme replacement theory. So they're using as an enzyme replacement theory as well as in a bone transplantation mechanism based on get cure this disease. in which functional ADA is used to their patient by the injections. So this total they this enter to give as how with help of in a injection only. But the problem was both in these approaches that they are not completely their curative. So when these both are we are giving, it is not they are permanently curative. But as a first step there towards the gene therapy, the lymphocytes from as in a body, in the blood out of as in a patient grow as in culture outside of the body. So first here the lymphocyte cells to get grow as in a outside of the their body. In this, the functional ADA, that is the cDNA using the completely retroviral vector. Here cDNA get act as what? The vector. It is in introduced into as in that lymphocytes. When now we are introducing it as a complete lymphocytes, which are the subsequently returned to the patient. So once we are there using as a retroviral vectors to develop as lymphocytes as in a outside of the body, then that enter the lymphocytes to introduce into as in a patient. However, these cells are not immoral. The patients requires a periodic inflammation such as in a genetically engineered lymphocytes. So what we are introducing, that is in what? Genetically engineered lymphocytes, means artificially we are introducing. However, if the genes are isolated from as in a marrow cells producing the ADA introduced into the cell early embryogenic stage, it could be their permanent cure. So once we are introducing it as embryonic stage, then what happened? It is completely cured for this disease. Okay, this is in a gene therapy. Remaining we discuss in next class. Thank you for watching. Please share and subscribe and like. Thank you.